Okay. Why is biology? We've had a bit of a disaster. Okay, so let's start on here. Hi guys, if you are joining me, I'm actually going live on my YouTube channel. So if you want to jump across to my YouTube channel, you'll be able to see that I'm actually going to go live there with my screens and do this slightly different. So if you are joining me here, welcome guys. I have decided to move over to my YouTube channel to not only do the sessions, but also it means that you will be able to actually watch them back again. So they will, hi, those of you, I can see some of you are now joining me. Yeah, so jump over to my YouTube channel. You can chat in the chat box on there, and I will be running the session as I did before, where I'll display screens, I'll write and show you what you can answer, but I feel like the YouTube means that you've got access to it later as well because I go for so long. So thank you for joining me. I am going to look at the paper that we were doing last week. What it says behind me is my advert for my intense course that I am running on the 9th and the 10th of April. Now, EO, if I remember rightly, you are year 12. If you're year 12, it is more aimed for year 13 because it's an intense course for the exam. So guys, if you just joined, oh, you are year 13. For some reason, I thought you were year 12. If you're just joining me, jump over to my YouTube channel because I'm going to do my display on there. Shadima, yes, it is expensive, £320. Actually, a lot cheaper than it would be for having one-to-one with me. And it is over 12 hours worth of tuition. So it is 7 days worth of 7 days worth. Let's try that again. It's 7 hours worth on each of the days. And it is intense based on me going through all the papers, looking at what's come up over the last couple of years and going through content. But I'm going to go on to the YouTube channel. So I'm going to kind of jump between you, but mainly aim here. So what I will do now is I will be looking at the paper that we did last week and carrying on from there. If you do have any questions, then please do ask away. But I'm going to start with the heart question as it says here. So remember, whenever I do these sessions, I'm trying to help you not only be able to answer the question, because for a lot of you, then what you're going to be doing is having access to these mark schemes. But what will happen is when you are given the question again, you're actually going to lose out if you're only focusing on memorizing that mark scheme. So do make sure. No, it's not 320 for one-to-one. Absolutely not. Sorry, guys. The course that I'm running here is the one which is a group tuition session. So it is limited on the numbers of people who can attend, but it is a way in which I will be making sure that you get enough content and you can go through to aim for that highest grade. What's the definition of homeostasis? So homeostasis is where you want to maintain your internal environment with very set limits. So you're going to have to detect any changes that occur away from those set limits, and you want to correct the response to bring you back down. So Okay, we're going to move on to the question. So if you're just joining me on here, guys, please do go and jump on my YouTube channel where I'm actually going to go through live papers with you on the channel. So please do check that one out. We're going to start with the valves and how they control the flow of blood through the heart. So if you're just joining me, please do go check out my YouTube channel. And that is where I am currently actually running the session. I'm moving away from the TikTok because then it has resulted in a few people jumping on and being a bit disrespectful. So if you've just joined me, please do check out my YouTube channel. I'm on there now. So you're asked to complete the table about the roles of the two valves in the heart. Now, whenever you do this, it's important that you do think about what the valves actually are and what they're needed for when it comes to the heart. So if you haven't checked out my YouTube channel already, Biology with Christine, what you will find is on there, I have actually put up the whole course and all the content already. So there's specific videos for you looking at these areas. So if we were to look at the valves specifically within the heart, let's get out some of my pictures. What you will see is that they are very much related to the fact that you have got valves in your heart where you will have either it be open or it be shut. Now, it's really important when you're looking at this that you're understanding that those valves are a way in which the heart can prevent the backflow of blood. So if we're looking at the flow of blood, then what you want to do is you want to ensure that the blood is only ever going in one direction. So to ensure that that is the case, what we need to do is we need to have it that they only go in one direction. So it is a case of, I like to think of them like swinging doors that can only open in one direction, and then they come back and they stop that backflow of blood. So for those of you who are just joining, please do jump on to my YouTube channel. If you would like to actually join my actual session for these, which are the free sessions that I run, I am looking at the biology club I'm going to run, so you can come along and that will help you. So if you need to know aerobic respiration, go check out my module 5.2.2 playlist on my YouTube channel. The questions that I'm actually looking at on the YouTube channel is going to be specifically on the topic area. So I'm live streaming that right now, and you can actually see. Hi, Dr. PCR. You can actually see the question paper, and I've also got my board up as well. So yeah, I want to try and help as many people as I possibly can. So if you are interested in joining the session tonight, then what you can do, there we go. Hi guys, I now see you on here. You did a heart dissection today. Nice, Shadima. How do you join the biology club? Well, the biology club will be a case of I will send out a link for doing emails so that you can then actually see what is happening with regards to on the weekly link for these sessions. So you can actually attend the live stream. So if you are interested in attending my Zoom lesson that I am running right now, copy the invitation. If you are interested, then if you click on that link, that will actually let you come to my session that I'm running right now. Where do you get the PDF for the questions? So the questions that I'm currently doing is 20 2020 paper 3. That's the one that we're looking at the moment. So if you are interested in joining, please do click on the link and you can come to the session. These are the ones that I run on a Monday and a Wednesday. I'm trying it over onto my YouTube channel now because I wasn't happy with how Monday's session went. So if you do want to join, then please do click on the link. But here we go. We're going to answer these questions. So it's asking, there we go, Anna, welcome. Hi, Alex. So we are now looking at this question. So you should hopefully, Anna, be able to see my picture and have access to it. Okay, so what is the source then of the blood? Now, the two valves, what we're looking at is the flow of the blood going in only one direction. So because we want it to only go in one direction, we are told that the valve that controls the blood flow is on the right semilunar valve. So we know it's a semilunar valve because he's already told us. Therefore, we need to understand if it's the semilunar valve. We're not talking about our atrioventricular valves. That's these bottom valves here. So this is my atrioventricular valve, and up the top is my semilunar valve. So what I need to understand is, as we're going through, we have it all to do with pressure. Yeah, Shadima, the link is on my YouTube channel. So guys, if you did want to join, please do jump on to my YouTube channel. And if you want to be in the live session as it is, rather than the live streaming, then you can send a click on the link that I've put on the chat in the YouTube channel, and it will take you straight to the Zoom session. So we've got this high pressure. Now, this high pressure is going to result in blood flowing from the atrium down into the ventricle. So therefore, there has to be a lower pressure in the ventricle compared to the atrium. Now, as I said before, what you've got is these swinging doors. So if you've got the swinging doors, thank you, Anna. I'm glad that you are finding it useful. There we go. So if you do want to join, then please do click away and you'll be able to see my session. You'll be able to see what I'm writing rather than live streaming on my YouTube channel. So those of you who are asking to join, please do, and I will keep going. Hi, Alex. Glad you're in here. So if you are joining me on my TikTok, I'm jumping over to my YouTube channel. And if you go on my YouTube channel and you want to join the live session, then you can click on the link, which is in the chat. And I'm going to continue the questions now. So with regards to the valves, the whole thing that we're looking at is about this pressure. Now, as I said before, what you've got is the fact that these valves are like swinging doors. Because they are swinging doors, they are going to result in you having this fact that the swingingness can only happen in one direction. So it can swim open, but stops when it gets to that point there. And because of that, what you then end up with is, so you've got this swing and stopping. That's then going to result in the fact that the blood pressure has to be higher underneath than it is above. So if you think about that and you think about the fact that we have this pressure difference for the valve above here, which is my semilunar valve, when it is shut, it has to be shut because of the pressure being greater above than it was below. So when I dissected the heart, what I then did is I actually had to move the valves because obviously when it's being dissected, it's been removed from the organism. There's no blood flowing through it. There's no filling up of the chambers. And so what you're getting here is you can see I have, looking from above, this is my atrium. So if I just add in here. 1 second. My atrium up here. So here is my atrium. And then underneath is the ventricle. So the ventricle is underneath, and you can only see a small section of the ventricle down there, and the atrium is above. Now, if I then look to move from my atrioventricular valve and I come from below this time and I'm looking up, what you're going to see is you can see quite clearly that I've got my, switch screens again. I have got on this side, this time, I'm coming from below. And because I'm coming from below, what that's going to result in, so my ventricle, my atrium is up here, and my ventricle is below. So if it's open, if my atrioventricular valve is open, that's therefore because that pressure above was greater than underneath. So when we look at the atrioventricular valves, they can either be open or I had to actually close them with my forceps. Now, remember I said you've got this swinging door. So if you've got the swinging door, what you've got is if the pressure is greater above, it's going to push those swinging doors down. When the pressure below becomes greater, that's going to push them back up to the point where they can no longer move because they have these strings attached to them. We call them the heartstrings, and they are going to stop it from opening, from flipping backwards. So you've got these swinging doors that can swing that way, but then will stop at this point here. Now, as they stop, that's because the pressure underneath has actually become greater than the pressure which was above. So what you've done is you have now closed that valve. So you can see in my diagram here, I've got a higher pressure and a lower pressure. Now, if you look above, what you'll see is my pressure in my ventricle gets greater than the pressure in my pulmonary artery up here. Therefore, that is going to open those valves. So these ones are called semilunar valves because they're like little pockets. Now, unfortunately, the heart that I was dissecting, I didn't get to see the semilunar valves very well. So I couldn't show this, but when I do it in lessons, I'll normally pull it out to show you that filling up of blood. So what you get is it all to do with the changing in pressures. The greater the pressure will push open a door. When the pressure gets lower, it will shut the door. So this is really important when we're looking at this, that we know that we have got our semilunar valve on the right side. Therefore, where was the source of blood? Well, the blood was in the right ventricle, and then the destination of the blood is going to be in the pulmonary artery. So then we look at the left side, and we know that with the left side of the heart, what you've got is the left atrium. So we've got our left atrium up here. We've got our left ventricle, and we're looking at this atrioventricular valve. Now, if your teacher's anything like me, they'll told you about the bicuspid valve. They may even have called it the mitral valve. You're really lucky in your exam that you just have to call it the atrioventricular valve. That's all they're going to ask for. They're not going to expect any more than the atrioventricular valve. So if we know we're in our left atrium, and we know the destination of the blood is the left ventricle, therefore, we would say that this is going to be the left atrioventricular valve. Now, I am writing in shorthand. I'm not going to write the full thing out. However, in your exam, please do write the full part out. Just need to know the label that's on the diagram. So with regards to the histology, they don't tend to give you the histology unless they're looking for the coronary arteries. They would be more looking for you to be able to label the chambers of the heart. and the blood vessels that are associated. So you need to know about your pulmonary veins, your pulmonary artery, your vena cavas, and your aortic arches. Am I allowed to have the camera on? You can, but we shouldn't be able to see it. Do, do, do. The heart is an interesting one. It's one of the questions that students, for some reason, do stress about. But one of the things that I would say that my recollection from the past exams that I've done, most students either know it or they don't. They memorize those chambers and think that's all they're ever going to be asked. It's not going to be as simple as that. So if they are going to ask you about the heart, if it's a really nice question, just completely checking your AO1, your recall ability, it will be name those chambers. It will be like this one, where is the valves? So therefore, where's the blood flowing from and to? But when it comes to those application questions, it's more about how that pressure changes due to systole or diastole and linking that to the neurons and how they then are going to have an effect on the pressure building up. So let's move on. Here we have the heart that's been shown to us, and we've been told that there's a ventricular septal defect. So they're telling you in the question that it's a hole in the septum. So yes, systole is a contraction. That is correct, Anna. So systole, whenever we talk about systole, we're talking about the atrial systole, the contraction of the atria at the same time, or we could be talking about the ventricular systole, the ventricles contracting. Diastole is the relaxation. So if you think about the heart as the chambers, you've got contraction, contraction, relax, contraction, contraction, relaxation. So it's basically atrial systole, ventricular systole, diastole. Atrial systole, ventricular systole, diastole. So you want to remember that you've got the first one, the atrial systole, then you have the ventricular systole, and there's a slight delay between the two. So it's important to note that they're never going to go at the same time. Guys, if you are joining me on the TikTok, I'm on my YouTube channel live right now. And you can join the session if you would like. The other thing I would say to you is if you want specific subject areas, go and check out my YouTube channel and look at the specific playlist, which is linked to your specification at the actual module level. What would you recommend doing if we run out of past papers for OCR? Okay, so you've got 2017 to 2023. I would say do AS papers. If you do the AS papers, what you'll notice is the AS papers actually have a lot of content that can then go into the A-level paper. Can everyone see in the YouTube TikTok? What do you mean by that? What, the YouTube channel is a bit laggy? Well, if that's the case, or are you saying my live session is lagging? Oh, the YouTube is lagging. Yeah, the YouTube's always running a little bit late, which is why, for those of you who are in my actual Zoom session, you should be having it live without any lagging at all. If there is issues in the actual Zoom, do let me know, guys, because that will help me. But you shouldn't be able to see on the YouTube channel. I can see Alex, but you shouldn't be able to see Alex on the YouTube channel at all. Hi, Alex, by the way. Nice to put a face to a name.
Hello.
Right, let's carry on with this question then and keep going. So they've told us a bit of information, and straight away they've told us in the question that there is a hole in the heart, and we can see that quite clearly here. Now we should know that the septum is about separating our two sides because we have a closed double circulatory system. So I'm not moving on yet. All I'm doing is I'm looking at this and saying, okay, yes, there is a Zoom random user. If you jump onto my YouTube channel, in the chat, there's a link you can click on to join the session. Yes, biology. With regards to diastole, then we do tend to talk about it with the repolarization. So if you are year 13, oh, Chidima, I actually see a picture of you now. Hi. There we go. Facebook.
Hello, hello.
So yeah, so with regards to diastole, when you're looking at your ECG, what you'll be noticing is you've got your P wave, your QRS complex, and then your T wave. The T wave is ventricular repolarization. So when we're going into the neurons, we'll then be talking about that in the fact of those repolarization of those neurons need to occur so that that relaxation of the heart can happen. Okay, let's carry on with the questions. And if you do have any questions, if you're in my session, please do let me know. And welcome to those of you who are in my actual Zoom session. Okay, describe and explain why people with VSD can easily become tired. So let's go back up and remind ourselves, we've got this closed double circulatory system is what we should be having happening. Now, the reason we want that is because we want to ensure that on the left side, we know that we're going to have our oxygenated blood that's going to be sent round into the circulation, into the systemic circulation. So it's going to go out all the way up to my head, to my fingertips, down to my toes. That is going to be that oxygenated blood circulating around. And what we should have coming back in to our right side is deoxygenated blood. So it's a closed system, it's a double circulatory system. Therefore, that means that there should be no mixing of our blood. And that's going to maximize our concentration gradients and therefore helping us to ensure that what we've got is this efficient exchange and transport system. So if we go down here where it says describe, and then it says to explain. Two parts to this question. Describe is one part, explain is another. So guys, if you're joining me on TikTok, I'm on my YouTube channel. Please do jump across there. You can actually join my live Zoom as well, which means you get it in real time rather than in a case of the lag.
Also, you get to show your camera.
Yeah, and you get to share your camera if you want to join the live. So I'm just reading the comment. Biology. I'm just checking who biology actually is. There we go. Caused by the SCN. So yes, so when you are talking about, it's key, Anna, to go back to what you've asked in your question, be careful what you're saying with regards to the fact that you've got your depolarization and your repolarization. So you want to try and use your key terms, depolarization, repolarization, hyperpolarization. So whenever we talk about the sinoatrial node, and whenever we talk about that being the excitation going across the atria, what we're talking about here is depolarization
What?
of the membrane,
Okay.
and that therefore allowing us to have the, one second, guys.
Did you just hear random voices, or was it just me?
No, there was definitely random voices, which is why I stopped to just double check what I was going to do. I want to make sure that everybody is muted except for me. There we go. Right. So moving forward to what this question was asking. So it's a describe and explain question. So describe what you see would be the reason. So you can clearly see they've given you some help in this question. There's a big hole. They've even told you there's a hole. So therefore, what's going to happen if we describe it? You've got the fact that the blood is going to leak from one side to the other. To your right and vice versa. So because that's going to have this leakage going from one side to another, that's therefore going to result in you having a concentration gradient that's not as efficient. So there's going to be less oxygenated blood that is going to actually be moving around the body. So because we are now mixing our blood, our concentration gradient is going to be affected. So we've got the blood that's being circulated around now is going to have less oxygen. That oxygen is required for aerobic respiration. So because that oxygen is required for aerobic respiration, you've got less oxygen because the mixing of the blood, that concentration gradient is affected. Aerobic respiration is going to be affected. So we've got less aerobic respiration, therefore less ATP production, therefore becoming more tired. So you can quite clearly see that it is describe what you see and then explain with your knowledge of why that would have an effect. Okay, let's move on then. So this next question is asking you about an enzyme, and students get questions like this and they freak out. And the reason they freak out is because they're not actually looking at the information that they've been told. How do you revise a topic you've forgotten in biology? Go back over that stuff. So check out my YouTube channel for the videos. How do you know it will leak? Well, if you've got a hole, like anywhere, the hole's going to let things move in and out. So therefore, because that hole is present, that's going to cause that leakage in both directions. It's not to do with the valve closing. So Paul, be careful on that one. It's not about the valves not closing properly because it's not the valve that's the issue. Because this is the valve. If I just color, I'll circle these in so you can see. That's the valve. Oh, no, I'll try that again. That's the valve, that's the valve, that's the valve, that's the valve. What they've shown you in the picture is this hole in between. So what we're seeing is my blood is going to leak in that direction and in that direction. So there's going to be this leakage across from the two valves. Oh, Lynn, I'm glad you're finding this better. I've been meaning to move on to this. This is basically how I do my one-to-ones and my group tuition sessions. And so therefore I was trying to find a better way. Is there any resources? Daryl, I'm in the process of actually building. I'm not finished the booklet yet, but there is one coming. I'm hoping by the end of this week, because I'm feeling much better than I was, it should be done. I need to do the chi-squared test, and then I just want to add in a few extra bits for you. Oh, I'm glad you find it better. Well, this is basically how I run my sessions and my business, so it should be better. Okay. Creating kinase then. When students get a question like this, they do tend to freak out and they seem to get confused. They're like, I wasn't taught this. My teacher never told me about creatine kinase. Well, creatine kinase, they've told you it's an enzyme. They've even told you here, if they hadn't given you the hint with it being an ACE, they've told you that it's an enzyme. And they've told you that it's an enzyme that catalyzes a reaction in the heart muscle. So already we know it's an enzyme. They tell us it is an enzyme that catalyzes the reaction in the heart muscle. So if you've got high levels of the CK, the creatine kinase in the blood, so hold on, it's in the blood, and it's going to indicate that a person may have had a heart attack. So how did it get in the blood? So we know that it is found in the heart muscle. We know that the muscle is an organ. An organ, oh my gosh, my writing is atrocious. Let me just make this. Hold on 1 second. I'm going to change my sharing a second. I'm going to remove this because we don't really need that anymore. And just go with this. And then I can make this a bit bigger. Oh, wow, that's gone really big on me. And then my writing should actually be a bit easier. Let's try that again. So the muscle we know is an organ. An organ we know is made up of tissues. And our tissues are made up of cells. So we're already being given some information in the question. These are exam techniques. These are where what you're actually learning about, all those bits coming together, they are relevant with those hints. So when the examiner writes these questions, they are writing them for you to have some hints if you know where to find them. So you've got the heart muscle. You know that it's an organ that's made up of tissue that made up of cells. Therefore, how is this indicating that there's a heart attack? Well, you should know that this is intracellular because you were told it is catalyzing the reactions in the heart muscle, the muscle being a cell. So therefore, if you understand that it's an intracellular enzyme, what you can then see is that if it is in the blood, if this enzyme, let's just remind ourselves about enzymes, they are globular proteins. So if they are globular proteins, they're going to be quite large. The only way that they can actually get out is if they actually have had some rupture of the membrane within the cell. So therefore, if you have the presence of it, that is going to indicate that there is damage in the cell. So it's an intracellular enzyme which is now located in the blood. Therefore, it has to have been released somehow, and that would therefore result in an indication that there is damage. Okay, so mice are often used in lab studies to research the treatment for the heart condition. They're often cloned. So if you are year 12, how did I know it was an intracellular? Because they told me it catalyzes the reaction, and the real indicator was that word in. Inside the heart muscle, the muscle is made up of cells. Now, the blood, remember, blood is going to circulate in a closed system. So if it is circulating in a closed system, that therefore means that they've given us the hint. Well, this is basically what I do this is an exam technique that you learn with practice the more you do, which is why I'm doing these sessions and now doing them on the YouTube channel, because you'll be able to watch these back whenever you want to. It's also, it comes with practice. You've got to remember, I've done 20 years of teaching, and I actually only fully understood the links when I taught the whole course all the way through. So the more you get into a practice of not focusing solely on, this is one subject area. I'm going to study exchange systems. I'm going to study the heart, for example. If you only focus on that one subject area, what that then does is you're putting things into compartments. And then that question comes at you. And this question is actually checking, do you know about proteins, enzymes? Do you know about membrane structure? Do you know about the fact that these can be intracellular or extracellular? So my next question would be, it's an intracellular enzyme, so where was it made? Well, it's a protein that was made through the process of transcription and translation. And therefore, if it's through transcription and translation, where were those ribosomes? Ribosomes have to either be free floating in the cytoplasm or they are going to be in the attached to the rough endoplasmic reticulum. So if we're talking about an intracellular, as this one was, that had to be the ribosomes, which are free floating. So again, it's that whole thing of you need to look at those links. I mean, that's basically why I built my YouTube channel and why I do these sessions for you, to help you to identify those links. Okay, moving on. So the next question then is about the mice and the fact that they're clones. So suggest why we're going to use clones for the use in these lab studies. So what are clones? So this is an interesting one, and this links to your module 6, cloning and biotechnology. So the clones, we know that clones are going to be genetically identical. So if they are genetically identical, what that therefore means is whatever conditions we give to them, we will be able to see the variety of the experiment as a way, because they're genetically identical. Now, be careful. A lot of you are saying controls, 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 but you have to talk about the fact that the genetic variation, i.e., there's no genetic variation, is what allows for it to be a control variable. It's not enough to say control variable. They're not going to give you the marking point by stating it's a control or control variables. You have to give more detail than that. You have to give them information that explains what that is. So why do we test on clones? Because therefore you are controlling the genetic makeup, because if there is a difference in the DNA sequence, that would therefore affect the potential primary structure, the amino acid sequence, that could affect the proteins that are being produced and therefore affect the shape. So you want to control that variable by controlling that they are genetically identical. So it's about giving them as much information as you can so that they're all going to have the same response, because you would expect, if they've got the same DNA sequence, that therefore will be transcribed, and that transcription will give us our mRNA sequence. And that mRNA sequence will then be translated with the anticodon, which is found on the tRNA, being complementary, and that will therefore result in us having the same primary structure of all of the genes that are going to be expressed. So how are mice cloned? Well, any type of cloning for your exam is looking at you knowing you can either do the somatic cell cloning or you can do embryonic twinning. So that's module 6 knowledge coming in. If they're going to ask you about the mice being cloned, they want to know, do you know those two different versions? Of course, Alex, ask away. That's the whole point in these sessions.
Okay. So what I'm wondering is we have a test in 6 days on it's on protein synthesis and enzymes. Do I need to know all the stuff about like transcription and translation and everything like that, or is it just like I don't know exactly what to know.
Okay, so module 2, protein synthesis, is nucleotides and nucleic acids. The
Hmm.
enzymes is that then linking to protein structure and the action. So if I was making the assumption, if they've told you it's protein synthesis and they've told you that it's enzymes, as a teacher, I would say to you, transcription translation could come up because it's protein synthesis, but also
Okay.
cell structure of the organelles involved in protein synthesis can come up. Also, you've got the enzyme action as to if you're looking at a competitive inhibitor or a non-competitive inhibitor or a substrate and an enzyme being complementary, that's down to protein structure. So it'd be all of them together, I would say. If you're going to revise transcription translation, absolutely. The cell structure of the organelles involved, RER, and then you've got your Golgi and your vesicles. If it is leaving extracellular, or if it's intracellular, then the ribosomes inside the cytoplasm. And then enzymes, the fact that they are tertiary structure, they're globular proteins. So they have a very specific shape, which was determined by the DNA. So it's a difficult one because if they're going to ask that any question on it, and I guess it's about a 45-minute exam.
Sorry. Usually they're 30 marks for around 30 minutes.
Oh, okay.
Yeah,
No, 30 marks is not 30 minutes, it's 1.33.
I know, but they're strict on timing in my school.
No, but that's what I'm saying. If they're strict on timing, they're not giving you enough time.
I know.
They're not showing you what it would be. So if they're giving you 30 marks and they're expecting you to do that in 30 minutes, they're actually doing you a disservice and you're not getting the time that you should be getting because that's not real what it would be like in the exam. In the exam, it's 1.33 for every mark. So every
Mhm.
mark times it by 1.33, that's how much time you should be allocated to that. So your teachers are doing you a disservice there. The one thing I would say to you is
I know.
if they're only asking you 30 marks worth, okay, they're
Mhm.
going to have to really open the subject up. So therefore, if it was 30 marks, I would be covering all of that within the questions
Okay,
that I'm giving you.
cool. Thank you so much.
You are very welcome. Is there a PDF of these questions? You can get them, as someone's already said, the OCR website. I'm doing, these ones are the 2020 paper 3. I jump around which paper I do, but you have access on the OCR website to all the exam papers up to 2022. I'm not sure if they've actually released the 2023 to you yet. I have access to them, but I'm not sure for students you have access yet. But 2022 is free, available for you to use with the mark scheme. So this one's 2020. And the reason I'm doing 2020, just so you know, is in my experience, most exam boards, when they do this, do a rotation for A level anyway in 3 years. So you had your 2024, 2023, 2022. So 2020, 2021 have been far enough away that you'll start to see some of those topic areas come through again. But don't just focus on those topic areas in those papers because they'll never ask those same questions again, would be what I would say. They don't ever repeat the questions. Okay, so just so to answer your question, they never repeat the questions, but they just make alternatives to it. So when I go through these sessions, I'll never just give you the mark scheme. You can do that yourself. What I want to show you is how they can go around this question to help you. Absolutely. I always tell you, will I be making predictions? Well, will I be making predictions? No, I don't believe in doing predictions. However, will I be using my research to guide my intense revision course? Absolutely. Um, so I don't believe in doing prediction papers because I know that there's lots of people out there who have already done it, and it worries me because some students are lazy. And they just want me to tell them what's going to be in the paper. And I could say that something's going to be on the paper, and I've guessed it, completely guessed it. And therefore, am I at fault for telling you that that was my prediction, or are you at fault for believing that and not doing revision around the whole topic area? So I don't like doing predictions. However, I have gone through and analyzed the papers. I have looked at what's come up. I have made a judgment myself, but then I've looked at every single paper and I've looked at every question that they have potentially asked on any point. And I have a lot of experience on this one as well. So the sessions that I'm running in Easter is a very intense session. It is basically a 7-hour day. So you will come online for 7 hours. Yep, it's intense. She might love that face you just built. 7 hours. And what's going to happen is I will be breaking it up into paper 1, paper 2, paper 3. And also what I'm going to do is you will have breaks throughout because I need to have a break and you'll need to have a break. But it is going to be an intense 7 hours, 2 days straight of everything you need to know from the course on application skills, course content, and also having the opportunity to ask questions if you have, like this, so that I can identify and help you. Because if you are paying for me to help you, I want to ensure you get the most out of your sessions. So do please, I love it. Someone's already jumped onto my TikTok and been like, come and check us out. We're a good tutoring service. There are so many good tutoring services out there, but do make sure that you research who they are so they're giving you what you need to be successful. Okay, last week, for the revision session, it's on the 9th and the 10th of April. So it's from 9:30 in the morning till 4:30 on those 2 days. So we did do question 3 on Monday session, but we didn't finish question 3. So I want to jump down because this is a very important one when it comes to you understanding statistical tests. Lynn, it is £320 for the 2 days, but it works out at about, I think it's £26 an hour a session, which is way cheaper than you would ever get. But you also have access to me afterwards as well. Anyway, back to the question. So for those of you who weren't here on Monday, we looked at this and it was to do with tropism, and it was to look at 2 different groups. And those groups were going to be 2 days in darkness or 1 day darkness, 1 day light. And then we got a set of results, which we analyzed last lesson. But what we're interested in is, oh, I'm going too fast, the statistical test. The groups, it will be group sessions. It won't be one-to-one. It will be group sessions like I'm doing right now. So it'll be like you are here right now. There is a limit on how many students I'm letting actually attend the session. So it's not like millions of students. I mean, it'd be amazing if millions of students wanted to do this, but that's not anything I would be able to give in a sense that you would gain the necessary improvements. Okay, so Anna, this is what we're about to talk about now, is how do we know which statistical test we're going to use? Which one do we want to use? So it's important that you know the different tests that you can actually do. Excuse me one second. So there are 4 tests you need to know about. There is the t-test. Now the t-test is either a paired or an unpaired. Now, whenever you're looking at a t-test, you're always going to be comparing means. And it's comparing means of 2 sets of data. So if I'm going to compare the mean of 2 sets of data, and it's that mean value, then I'm going to do a t-test. Now, whether it's a paired t-test or whether it's an unpaired t-test is whether they are in the same individual. So for example, if I were looking at exercise and I was looking at somebody who was checking their heart rate before they have done an activity, and then they do an activity and they check their heart rate again, that would be looking at the mean value of the same individual. Therefore, it would be a paired t-test that I'd be looking at. I'd be looking at the difference between before and after. If I'm looking at two completely different situations, so if I was looking at, for example, in this case, I'm looking at A and B, two different groups, group A, group B, and I'm looking at the mean values of A and B. I want to compare them. Group A got 2 days of darkness. Group B got 1 day darkness, 1 day light. They are not linked at all. Therefore, that would be a unpaired t-test. So in this question, they'd already told us we were looking at means and we were looking at two different values. So therefore, I would want to do a t-test because I'm comparing means. Well, this is the thing with null hypothesis. It always comes through. So we always want to do a null hypothesis, whether we're doing a t-test, whether we're doing Spearman's rank, whether we're doing the chi-squared, it's always going to be a null hypothesis that there's going to be no difference between the two. So how do you know if it's a Spearman's rank? So a Spearman's rank would be if you have the variables where there is going to be some form of a relationship. So if I have, for example, the number of ice cream sales and shark attacks, I love doing that one because if I'm looking at number of ice cream sales and shark attacks, I would find that there's probably going to be a positive correlation between those two. But if I want to be sure on that, I want to look at ranking the data and then finding out whether there is a positive or negative correlation. So if there's a relationship between the variables that I'm looking at, that would be a Spearman's rank. So if one is correlated with another. I'll come back to the degrees of freedom in a minute because it is different depending on which one we're looking at. I just want to finish on the other question that you had with how do you know if it's chi-squared? If it's chi-squared, which they're going to ask us later on, it's about us understanding that to do chi-squared, we need to have an expected value. So we need to have a way of working out the expected value when it comes to chi-squared. So degrees of freedom then. When it comes to degrees of freedom, if we're doing the Spearman's rank, you need to look at the number of paired sets. So you do your n. So if I have, for example, 2 sets of variables and I have 7 ranks in those variables, so I've got 1 to 7, I would therefore have my n equals 7. So my degrees of freedom would be n equals 7. If I'm looking at the t-test, then what I want to do is I want to have my n of A plus my n of B minus 2 to work out my degrees of freedom. And if I am doing my chi-squared value, then it's the number that I have got minus 1. So the categories that I've got minus 1. So if I'm looking at, for example, a ratio which goes 1 to 1 to 1 to 1. I've got 4 possible categories. So 4 classes minus 1. So therefore my degrees of freedom would be 3. So they would give you some information in the exam. So if we actually look at this one, because it's going to get us to do a chi-squared or expect us to evaluate a chi-squared, what I have is I have away from light, neither away nor towards, and towards light. So 3 sets of data recorded. Now that's 3.2. So just to go back up and show you what 3.2 looked like. This was table 3.2, and we had different data with regards to whether they grew towards or away. And when we are looking at the fact that they are in the stem and in the roots. So if these are our observed bits of data, we would then need to work out our expected ratio. So if we've got a null hypothesis, There is no difference between the expected direction of root growth and the observed direction of root growth. We had 3 classes. Just to go back up a second. So we had away, neither away nor towards, and towards. So we would add the total together. So what's that? 30, 60. That's right, isn't it? So we had 29, we had 20, we had 11. So therefore our total was 60. We have 3, so we would literally do 60 divided by 3, which would give us 20. We would have expected 20 at each. Do you have a playlist on these tests? Not yet, Anna. Anna, I've been doing the workbook so that I can then do some videos to go alongside the workbook on specifically these key skills that people need. So they are coming. Just bear with me on that one. Need to know the equations. No, the equations will be provided, Paul. So they will give you the equations. The only equations they won't give you are things like the percentage change, the genetic polymorphic, what am I trying to say here? Polymorphic, what am I saying? It's in the genetic biodiversity, so as you can see, my brain's not working quite. Loci, thank you, polymorphic loci. And things like the no, they will give you how to work out the volume of a cylinder because that's in oh, percentage uncertainty. When's your test? Is it before Monday?
It's in 6 days.
Yeah, but that's before. So if it's in, so it's on Monday. No, Tuesday.
Tuesday, yeah.
Tuesday. So Monday session. Send me an email to remind me on Monday, and I will do that. Percentage uncertainty. Yeah. So they will expect you to understand how to work out uncertainty. So if we go back up, just because that's part of your practical skills, and this is something they'll do. They've told us that we've got our millimeter as being, hold on a second. There we go. Excuse me. Our mean length was measured in millimeters. So therefore, I can make the assumption they've used a ruler to measure that. So I therefore know that my uncertainty in that data is plus or minus 0.5 of a millimeter. So it's normally when they're looking at uncertainty, it is either given to you in the question or they will give you information for you to work that out. And they'll expect you to know that it should be 2 times the uncertainty divided by the quantity measured. And that will give you times 100 your percentage error in your uncertainty. I would say lots of practice. So if you can do as much practice as possible, that will help you with regards to the maths questions. It is just about practice makes perfect. The more you do, the easier it becomes. Once you get to the point of if you've got loads of, for example, statistical questions, you just know how to answer that. Okay, so going back here, there is no difference between, so our null hypothesis they've given to us. So we're going to figure out whether we want to reject or accept this null hypothesis based on the statistical test. So they've told us that we've got a chi-squared value that's being calculated, which is 8.10. So they're either going to ask you to do that yourself by giving you the equation, or they'll give you the value that's already calculated like they have done here. They then want you to go to the table to look at this and say, right, well, we've got 0.05. We always go to the probability of 0.05. So what we're saying is that 5% chance in the uncertainty in that data, we are 95% confident if we get that value. So when we look at this one then, when we're looking at our numbers, we need to understand that our degrees of freedom is our classes n minus 1. We had 3 classes minus 1, therefore our degrees of freedom is 2. So if I look at my 2 at 0.05, the critical value is 5.99. So now what I need to do is I need to say that my 8.10, my calculated value, is greater than the 5.99 critical value. Therefore, because my 8.10 is greater than 5.99 at the 2 degrees of freedom, that means that there is a significant difference at a p-value of 0.05. However, if I look at this one, let me just change my color. If I look at this, my 9.21, I know that my 8.10 sits in between here. So I'm 99% confident in my data. I'm not 99% confident. Paul, that's the one that I'm working on right now. So the stats pamphlet, as you say, booklet, it's in the process. The hope is it'll be done by this Friday. So I've got time tomorrow in between my clients, and then I've got Friday. So that is the plan, that it'll be done hopefully by Friday. If not by Friday, by the end of play on Monday. That's the time limit I've given. Monday and Wednesdays are the days that I do these sessions. With regards to this. Right. Back to the question. So 8.10 is greater than 5.99. Because it's greater than 5.99, I'm saying that there is a significance. So there is a significant difference. Shadima, to answer your question, I will do this until probably about another 10, 15 minutes. And then I'll stop for tonight. So there is a significant difference at a p-value of 0.05. However, not significant at a 0.01 p-value. So that would be the way in which we do this. You're very welcome. And so therefore, what we're saying is at the p equals 0.05, we are going to reject the null hypothesis. If the number was lower, so if my number were somewhere in between here or over here, then I would accept my null hypothesis. Because it's away, I am going to reject the null hypothesis. Could you quickly talk about the structure of 9-mark questions? My teacher's predicting they'll come up. Okay, they could go up to a 9-marker. They haven't done a 9-marker in a long time. It tends to be more 6-markers, but if they are going to do a 9-marker, it will be probably a 3-part question. And what I mean by that is that if they're going to do a 3-part question, it'll be describe and explain with the validity. I don't think a 9-marker is going to come up, to be honest. They did stop them when they started the 2017. I haven't seen a 9-marker since a 2014, 2015 paper. But there is a part that says in the specification that it could go up to a 9-marker, but I don't think it will. But again, I might be wrong in my predictions and your teacher might be right. And that's why I don't believe in predictions. I find that predictions can only work so much. So yeah, you've just got to be careful on that. Right. What can the student conclude? Well, there you go. We've just already concluded it above here. Because it's greater, 1 marking point, 2 marking points, 3 marking points, 4 marking points. Well, that's just a way. I mean, it's asking for a 3 marker, but I tend to find that whenever you look at validity on this one, if the answer has given you towards, so we're saying it hasn't reached that point there because it's less than the 9.21, the critical value, that therefore means that we are not 99% confident. In fact, this is 99.9% confident. We are not 99.9% confident in our data. We're only at the 95% confidence level. So it's just another marking point that you could give them to show you know how to write a conclusion based on the results that they've given you. That is a solely application question. Okay, so moving on this one then. Solution in which the seedlings were grown contained various dissolved ions. Now, again, I'm going to jump around here slightly. Dissolved ions, right. Well, they're dissolved. So what's that talking about? We're talking about water and we're talking about the solvent property of water. So therefore, I love that. Someone was going to actually try and mark it for me. If we look at this one, supplies the elements that form part of the structure of amino acids and nucleotides. Absolutely. It's looking for you. However, Chidima, be careful. You went and you said it was ammonium. Now, this is an interesting one because ammonium ions are not good at actually being utilized in the production of amino acids. And this is because you are, don't worry about it. This is because you're year 12 and not year 13 yet. You haven't covered this. So in year 13, you're going to learn about the nitrogen cycle and you're going to learn about the fact that it's the nitrate ions that are required for that incorporation into the ammonia and the nucleotides. So it's the nitrate and not the ammonia. However, when people sat this exam, what they realized is, well, technically, some organisms can actually incorporate ammonium ions in, and they did accept it. So it doesn't mean you won't get the marking point, but
Yeah,
it is
because
important.
I had like a mock and a similar question came up and I put NH4 plus and I still got the mark. So I was
Yeah.
just wondering.
So this is why. So what I want you to understand is that you'll get the marking point if they have made an agreement in that exam that they're going to accept it. So it could be that the mark scheme doesn't allow ammonium ions because they solely want you to write nitrate ions. So it really depends on the examiners that are marking it, and also it depends on what they want you to have gained it from. Now, you're being told it's plants because you're in seedlings. So therefore, they will accept that. For the plants, the ammonia as well as the nitrate ions. But what they actually want you to write is nitrate ions. But the year that they sat this in 2020, there was enough students who wrote ammonium ions. They then allowed it on the exam paper. So you'll notice when you look at mark schemes where it says allow, that's the bit that they allow because there has been a majority of students and they've therefore gone, do you know what? Actually, we will allow it, even though it wasn't the answer we were looking for. Okay, moving on, and you correctly did right. That was very difficult to write this in the exam, in the chat, but do make sure in your exam that you are writing your numbers in the right place. So obviously if you do chemistry, as Chidima does, I know that you do, it means that you know how to write the chemical formula If you write this wrong in the exam, if I were to write PO4 3 minus and my numbers be too big, as an examiner, you're not going to be allowed that. So it's really important that you understand that you need to write clearly. My writing is atrocious, and I know it's atrocious. However, if I'm going to write in an exam, I'm going to take a bit more time. So do make sure things like that you are taking the time.
Doctor's
Uh,
handwriting.
Sorry?
I said doctor's handwriting.
Oh, I know. Joking, right? But the thing is, we get so lazy now. It sounds ridiculous, but we're so lazy at writing because we type everything, because we do things digitally, that actually for now I do so much on here and I'm trying to write really quickly. It does mean that my writing is bad. And I have to say, I get a snapshot of, so if the way I'm writing this, and you're right, that is basically what you can see. I see as an examiner, I get a photograph of your written work. And therefore, because I get a photograph of your written work, if your writing is atrocious and I can't read it, I can't give you the marks. So if you are someone who struggles with your handwriting, if they haven't already identified that as being a problem and given you a normal working way of working, which is through using word processing, and the exam board have to allow this, then you have to take the time when writing your oh, that's hilarious. When writing your answer. So please do take the time. Okay. In the investigation, students controlled light, temperature, and mineral concentration. What other factor should be controlled? This is a very much module 1 question, by the way. A lot of people are like, what do you do about practicals? This is a module 1 question because it's asking you about controlling an investigation. So control variables. So therefore, you need to understand if you're going to be controlling those variables, absolutely. How would that have an effect, though? How would soil pH have an effect?
Uh, like wouldn't it determine um like the rate at which the plant will grow at? I don't know if that makes sense.
But the question is about other factors that should have controlled. How would pH have an effect on the growth?
Because would the plant not grow differently based on the pH?
But why? What's pH got to do with it?
Affect the enzymes in the plant?
Potentially, yeah. Because the enzymes are needed for the plant to grow. But hold on a second, I want you to go into more detail now. You said enzymes, and the pH is going to have an effect on the enzymes. I want to know more. How does the pH have an effect on the enzyme?
Uh, affects the charges on the active site and the ionic and hydrogen bonds in the tertiary structure, which
Nice.
change the shape of the active site. Yeah.
Absolutely. And that's what I want you to know. For those of you who are saying denaturing the active site, it's not about denaturing the active site, it's about denaturing the protein affecting the tertiary structure. So by the pH, those hydrogen ions interacting with the bonds which formed the tertiary structure, you are therefore affecting the way in which it will be able to function. She's year 12. Nailing it though, Shadima. Great answer. Great answer.
I was
Um,
gonna say, even I don't know that, and I'm here 12.
well,
Thank
Chidima does come
you.
to my sessions every week, so
Gracias. Yeah.
but to be fair, you have been hitting consistently really high grades, right?
Oh, yeah.
And that's because you're putting the effort in little and often, or lots and often, because I'm here for like an hour and a half every week. And actually 2 weeks. So you're doing, in a sense, every time you come to my sessions, that's 3 hours of tuition you do every week.
I
But
never
it's
thought about it like that.
exactly, it doesn't feel that way, but that's what it is. When you come to a session, that 1 hour a week that you do, or in the sense of, because I always jump on here for an hour and a half anyway, that therefore has given you that 3 hours in 1 week. Now, if I jump on at another point, that's an extra bit as well. And if you're watching any of my videos, you're doing more as well. And that's the thing, like when I looked at the YouTube channel and I saw how many hours were actually being watched on my YouTube channel, I was like, that's mental that there's like 700 hours that have been watched. So if that was one student, not realizing that is a lot. Like if you think 24 hours in one day.
It's probably
Oh,
me
there
doing
we
my
go.
pre-reading. It's
But
probably
this is
me.
the thing. That's what I built them for. I built them so that you can go and actually watch the videos again, again, again until you get it. The more you do it, the more you understand. For the 2-day course, ideally, I have limited it to 30 people maximum. I do what I do a normal class size would be. So that'd be 30 people maximum on that one is ideally what I would limit it to. But do you know what? I'll be happy if I get 5 people. If I get one person, I'll still run the course. The reason being is it's about the efficiency of what I can offer you, as well as in a way of, because obviously I planned the sessions, the hours, the money that you pay isn't just for me being present and you being present in the session. The money you're paying for is also all the hours I've put in to plan the sessions, and you get the recordings and the resources afterwards. So oh, is there a due date to pay by? Well, until the spaces are gone, then up until the point of the day before, then that's what it would be. Those who come and pay obviously get their slot confirmed. So up until the point until all the slots have gone, then you can pay at any point until obviously the day of the session being run. Okay. Oh yeah, I'll finish on this one. I actually really like this question. It's the only question I've ever seen. I knew there's a reason why I picked this one. I love this question because this question is very much an application question about plant response, about the process of the geotropism. Now, you will have learned about phototropism. You'll have learned about auxin and the fact that the auxin can diffuse away from the tip, and you'll learn that the auxin is going to bind to a receptor. It's going to affect the plasticity of the cell wall, and therefore the cell can take in more water, it can elongate, it can get bigger. So you should know that already. And then they completely throw a curveball at you and give you something you've never experienced in your life and your teacher hasn't taught you. And you go, what on earth is this? Everything you need to answer this question is in front of you in the question. This is where you take the time and you read the information. So what have we got? We're being told that the position of the root, we've got our gravity coming down on both sides, but we've got our position of our root being horizontal and vertical. So we've got different positions of the root. We're then being told that we have an activity of a stathocyte. What's a stathocyte? I don't know. I've never heard that before. Well, they've told us up here it's a specialized cell, and it is the one that is going to control the growth of the plant root. So it's important you read the information carefully. Whereas most students do exactly what I did. They jump down to this picture and they see these words and they go, I just don't get it. That's not helpful. Take your time, work your way through. So one hypothesis for how a stathocyte let's try that again. Statocyte, this is where my dyslexia flick everything around the wrong way. I say things completely wrong. The statocyte controls the root growth involving small organelles called amyloblasts. And we're being shown these in our diagram. So we have these falling amyloblasts. So we can see that they're falling. We have our endoplasmic reticulum. Ooh, and we have calcium ions. If we have the root position down, we have gravity in that position. the amylioplasts are going to fall down. If they bind to the endoplasmic reticulum, they will therefore cause calcium to be released. The growth will continue. So let's have a look at what happens in the next diagram when we put the position in a different way. So when we have the root position coming in that horizontal direction, we still have gravity pulling down. Therefore, these amylioplasts are still going to fall. Because they're going to fall, they're going to bind and hit that endoplasmic reticulum. That's therefore going to cause that release of calcium ions. That therefore is going to cause the root to grow. So therefore, what can we conclude? Well, exactly that. We can conclude that these amylioplasts are going to be heavy. And the reason they're going to be heavy is because they're going to fall down. So the amyloplast is heavy, so we can assume that it's quite dense in the fact that it is going to fall down. When that amylioplast binds to the ER, what's going to happen is calcium ions will be released. So what can we then assume? We can assume that calcium ions are needed to stimulate the growth. And we can tell that because the root grows and it grows to the side where those calcium were released. Now it's a 2 markers, so I don't need to give all that information. I could get 2 marks from here. I could get 2 marks from there. I could get 2 marks from there. So it's important that when you look at these types of questions, that you actually just take some time to read that. Absolutely, Paul. I completely agree with you on that one. You don't need to know how the calcium ion stimulates the growth. You don't need to know that. They haven't asked you to explain it because it's not necessary. But if we think about it, the calcium ions that are being released are probably going to bind to a cascade of enzymes inside, which are going to then cause the reactions which allow for the growth to occur. But you don't need to know that. They won't give you a question that asks you for something if they haven't given you the information in front, unless you must know it. You don't need to know about calcium ions unless this was a muscle cell. Then you would need to say, well, the calcium ions bind to the troponin, which changes its shape, which therefore causes the tropomyosin to move out the way. That would be a, you must know that because you are therefore expected to have learnt that when you did the sliding filament theory. You're not expected to know this for geotropism. What you are expected to be able to do is apply knowledge to what